Hello, I'm David Brown. I'm the chair of the International Neural Networks Society College of Fellows. And with me today is Dr. Christof Koch. Dr. Koch is an American neuroscientist, best known for his work on the neural basis of consciousness. He is the president and chief scientist of the Allen Institute for Brain Science in Seattle. He received a Ph.D. in science for his work in the field of nonlinear information processing from the Max Planck Institute in Tubingen, Germany. He worked for 4 years at the Artificial Intelligence Laboratory at MIT before joining in 1986 the newly started Computation and Neural Systems Ph.D. program at the California Institute of Technology. And from 1986 until 2013, he was a professor there. He has authored more than 300 scientific papers and 5 books about how computers and neurons process information. He writes also a popular column, Consciousness Redux for Scientific American Mind, on scientific and popular topics pertaining to consciousness. Dr. Koch, I'd like to begin by asking, how did you get into the field?
Well, first of all, David, thank you for having me on this show. And then secondly, I need to correct a small infelicity. I used to be president of the Allen Institute for Brain Science, so it's a 350-person organization funded by Paul Allen, the Microsoft co-founder. But early on during the pandemic, I stepped down. This was planned. I stepped down and I'm now leading a smaller group, which is 70 scientists focused on brain observatories. So just for the record. I got involved because I grew up with books about physics. My heroes were, Einstein and Schrödinger and Heisenberg and Max Planck and the like in Germany, as you can probably tell by listening to me. And then I met, I was interested in becoming an astrophysicist. And then I met the brain. I encountered brains in an autopsy course. So I managed to participate in an autopsy course and was fascinated by the brain. And at the same time, this was the early 70s, I had constructed my own computer at home. I was a nerd, and I built my own computers, and I was fascinated by this relationship between information processing that the human brain is capable, or any brain is capable of, and the processing power of computers. And so I thought, like many people at the time, about networks of neurons and how they compute in the brain and how they could compute in computers. I obtained my PhD, as you mentioned, working on what's called the dendritic tree. That's the input end of neurons. They're very common. So it's like these extended filaments that extend from the cell body. And in some sense, in fact, recently Idan Segev in the Hebrew University have shown that you can model the processing in this dendritic tree where the synapses terminate from previous neurons terminate. You can itself model as a simple multi-layer network that then converges onto the cell body, where finally the action potential is generated that then propagates down the axon to the next neurons. And so I worked on how could information be processed in such dendritic trees. And then when I came to MIT, I joined the AI lab. I was further fascinated by how high-level operations like vision, particularly like where you decide to focus your eyes, where you shift your attention, right? I can look ahead, but I can independently focus my attention to different places in the visual field. How this happens, and then together with a scientist called Shimon Ullman, we came up with what at the time was enormously popular, this sort of saliency map hypothesis. The idea that there's a map inside the brains of humans and other creatures like monkeys and cats that represents in a topographic map what's of interest outside, both in terms of bottom-up factors as well as top-down, given the behavior of the animal. And then you visit sort of, the brain visits with moving eyes or shifting attention, visits the most salient location, then the next most salient location. So it travels around from location to location. That was enormously popular at the time. So that's how I got into what's today called computational neuroscience. There's a funny story there. When I graduated, my advisor, Tommy Poggio, at the time had moved already to MIT and couldn't join for my PhD defense. So he sent a telegram. You still remember that? Old technology?
Yes.
And he sent a telegram congratulating me, but he was also worried because at the time there was one other modeler in the United States of America looking at dendritic trees, Will Rall at NIH. And he was worried whether the field could support two people thinking about theoretical neuroscience in neurons. This was back in 1982. Yes, how things have changed. And then from there, I joined this through John Hopfield, the Hopfield of the Hopfield network. He was doing a sabbatical at MIT, and we shared an office. And then when Caltech started this new program, that at the time the first, that was fully dedicated on teaching students about about computer science and how computers work, and about brain science, and then getting them to think both about computation in brains and computations in computers. I joined that program back in '86, and then I was there 27 years.
What do you believe are your most significant accomplishments over this very productive career?
Well, so at the biophysical level, I've published literally hundreds of papers on how neurons compute, how they multiply, the function of the extracellular potential, right? Anytime you stick a piece of wire into the brain, you can get an electrical potential relative to ground, and it changes, and you can see ups and down brain waves, and you can measure those in the EEG. How do they serve? Do they serve to synchronize other neurons? So there's a lot of very rich biophysics there that we did models and experiment. Then the saliency map, that's probably the vast. So, I've on the order of 150,000 citations, very large number of them relates to this saliency map early on for vision, both biological vision as computer vision. Then I worked for many years with a neurosurgeon, Itzhak Fried at UCLA. And what he has to do in patients that have certain types of severe epileptic seizure, in some of these patients, depending on the type of seizure, if you do surgery to remove the foci of the seizure, you can eliminate or dramatically reduce the incidence of seizures. In some of these, you don't know from the outside where the foci is. So what you do, you implant electrodes into the brain, and then the patients stay on the ward for a week or two, may have some seizures, and then you can sort of triangulate from those implanted electrodes. That gives you also a chance, an opportunity, with their permission, of course, to record neurons in the living human brain as a person watches TV or looks at images or whatever. And that's where we discovered what's now known as Jennifer Aniston neuron, because one of the first neurons that we published on, this is all in Nature, was a neuron that responded selectively to very different views of Jennifer Aniston, which an American actress. So it turns out that we do actually have what people before sort of derided as grandmother neurons. For those things that you're very familiar with, your family, your friends, your loved ones, your office colleagues, your politicians, your celebrities, so people you see either in the flesh or on TV, on a screen all the time, or your dog or your keyboard, things that you see over and over and over again, your brain wires up through learning, and it can do it very quickly. We have evidence this can happen in a day. Particular neurons that respond to that. They're distributed. They're not all in one location. They're distributed. So if you think about your grandmother, or if you hear her or dream of her, or see her, in the flesh, as it were, there will be neurons that specifically respond. In fact, we even found neurons that respond to the letters Jennifer Aniston, or we have Bill Clinton neurons, etc. It just depends on what material we show to the patient. So that's pretty cool, because it was very unexpected. This was very unexpected. And then my best known contributions probably pertain to consciousness. So I worked for many years for 14 years with Francis Crick, who after he did his molecular biology work in the old world, moved to La Jolla and was very interested in understanding how the brain can give rise to consciousness. Because if you look at the foundational equation of physics, quantum mechanics, relativity theory, there's no consciousness there. You look at the periodic table, no consciousness there. You look at the endless ATGC chatter of your gene, no consciousness there, yet here we are, we're highly conscious. And so we focused on what we called, or what is now universally referred to as the neural correlates of consciousness, sort of the footprints of consciousness in the brain. And what happens in patients when they don't, when they have a disorder of consciousness, when you first see these footprints in a fetus, in a newborn, what happens in the late stage of life in dementia, how far can you track these footprints, in monkeys or in mice or in other animals, who else has it? So it gives you a very practical, operational, instrumental approach to understand something about consciousness. That now, 30 years later, leads to things like, building a conscious meter to detect the presence or absence of consciousness in patients. So that's what I've been doing. And then since I came here roughly 10 years ago at the behest of Paul Allen, we started this 10-year plan where he roughly invested $1 billion. And as I said, we have 350 scientists and engineers to lead a small number of very large projects. Really two projects. One is a cell type to try to, the neurons, unlike in a neural network where you have one type of neuron or two types of neurons, in the brain, they're probably on the order of 1,000 to 2,000 different types of neurons. Not just excitatory and inhibitory, but there are dozens of different subtypes in each area that differ by the genes they express, by the way they look, where they send the information, how they fire, do they fire in burst, in what layer they are, etc., when they develop, etc. And so we did a complete catalog. In fact, there was just a treasure trove of 17 papers in Nature, just published last week, of which we contributed 5. The Allen Institute contributed 5, laying out these different types of cells in a human, in monkey, and in mice. And of course, they're quite similar. They're not the same, but they're quite similar across these different species. So that's one big project we've contributed heavily. And then the other one, we built what we call these brain observatories. We published several papers in Nature about it, where we can record. It's a little bit like an astronomical observatory, where we can record on a highly stereotypical condition, highly reproducible condition, using professional staff, so not grad student, postdoc, but professional staff, where we can locate thousands, and this last paper was 100,000 neurons, precisely including a spatial location where it is and where we find these neurons, and make all this data available. In fact, we've now moved to this what's called open scope, where people can apply for experiments that we would run their experiment in our mice, using our equipment and our staff, run the experiment, generate all the cellular data, and make the data available in a standard format on the cloud. So now they can just analyze it. So it's really moving towards an observatory model of the sort you have in biology.
Yes, on my bedstand right now, there's a book which you wrote not very long ago, Amplifying Your Views on Consciousness. I'm sure, I know that's a focus of your attention. Looking forward, what do you think you'll be doing?
Well, so there's a lot of stuff to do here, practically speaking. So of course, I mean humans have thought about consciousness since, Aristotle first wrote about it, De Anima, 2,400 years ago. And literally every day there are new philosophical proposals coming out, but that isn't going to lead to progress. What we need is concrete, once we have an operational measure like the footprints of consciousness, tracking it, when does it happen, what happens in disorders of consciousness, who has it, etc., etc. So right now I'm involved, spending a lot of time with a large clinical trial with universities and companies, building what's called a conscious meter. So this is a device that would test whether, for example, a typical patient is let's say car accident, comes in, is unconscious, on life support, maybe for many days already, and is clearly alive, may occasionally move or groan or something, but you don't know whether he or she is conscious. You ask him, are you conscious? Do you hear me? If you hear me, move your eyes, move your finger, etc., but there's no response. So there are two possibilities now. Either the patient is truly not there, they're unconscious, although some brainstem reflexes are still there, or they are conscious, yet they are sort of, because of the acute brain trauma, are unable to respond. And this makes a huge difference, not only to the loved ones, because they want to know, is dad still there, but also to long-term care, because one of the big, there's this issue of withdrawal of care. When you're being told, well, your dad isn't there anymore and you won't come back anymore, then very often, due to all sorts of consideration, they withdraw care and the patient dies. If we could measure that and give people not only a diagnostic, but also a prognostic, where we can say, well, dad is actually in there, although he can't respond right now due to his injury, but there's some chance that he will actually recover at least some of his capabilities. It makes a huge difference. And so we're building this device. Essentially, you knock the brain using a magnetic pulse that goes through the skull, that excites the underlying neurons, and that triggers sort of this electrical reverberation that crisscrosses the brain that we can pick up with a high-density EEG, and then we can look at the complexity of this. So this is something that comes directly out of a theory of consciousness called integrated information theory of consciousness. So, there are a million of these patients, 1 million roughly worldwide. So this is not all that rare, this condition, for instance. So having such a tool would mark a real step of progress on this long road, historically long road, to understand the mind-body problem. So that's one thing that I'm involved with. Another large-scale collaboration is what's called an adversarial collaboration. This is a collaboration where you take the two most popular theories of consciousness, which is integrated information theory and global neuronal workspace, and they make some very conflicting predictions. So here the challenge was get these people together that usually don't like each other so much because they have opposing points of view, get them to agree on an experiment that if it comes out this way, it'll support IIT. If it comes out that way, it'll support the other theory, GNW, and then actually do it using different modalities in human, in normal, neurotypical subject, in patients, in non-human primates, and in mice. So this also involves 14 labs, in Europe, here, in China, etc. So it's another big collaboration, but very, very fascinating, because here you're touching things that are very close to us.
Yes.
Consciousness.
Now changing gears here. What would you remark about the early days of the field, when the International Neural Society was first formed?
Yeah, I mean, it was a very exciting time. I remember the first talks by Bernie Woodrow, by Stephen Grossberg, by John Hopfield. We had this series of meetings, first in Santa Barbara, then in outside Denver at Snowbird. Then they later more at Terry Sinofsky, Jeff Hinton. Then these later morphed into NIPS that ran for 20 years, and then that still exists. It's now called NeurIPS, and it's now migrated to either Whistler here in BC, Vancouver, or in Montreal. Well, there was this tremendous excitement because we felt we now finally had a beginning of a tool, these neural networks, these very simple structures. They were just 3 layers: input layer, hidden layer, and output layer, that could begin to address some of the problems that the previous generation, that stymied the previous generation of simple networks, i.e. the perceptron. And of course, in hindsight, we were deluded. It was way too early. Our computers were incredibly primitive. I remember when I came to MIT in 1982 to the AI lab, it took me, an hour to take one image, 256 by 256, and convolve it by simple Gaussian. It took me like an hour just to compute this one thing. So obviously, Moore's law has been incredibly good to us. Moore's law both for processing speed as well as for memory. And so now we can do these fantastic things where we can have a network that's, 20, 30, 40, 50 layers deep, and we can train it with thousands, tens of thousands, hundreds of thousands of hours of training data, and we can get it to do amazing things like, AlphaGo to play Go better than anyone else and be creative, to solve the protein problem, a long-standing problem of trying to predict the three-dimensional shapes of proteins from their one-dimensional nucleotide sequence, to speak to us, to understand human speech. So now sort of the foot is on the shoes on the other foot, in a sense. What can these neural networks, if they're given enough training data, what is it they cannot do? And once again, one really has to emphasize here technology. Scientists routinely overestimate the power of ideas and underestimate the power of technology. You see it in astronomy all the time, with the radical discontinuity in astronomy between, after the invention of the telescope and before. You see it, the invention of microscopy. You see it with, sequencing, sequencing, the genome project. So this is all driven by methods and by tool development. Here, what you got to remember, it was only in 1971, that's 50 years ago, that Intel introduced the first integrated personal computer that had 2,000 transistors, 2,000, okay? Fast forward to 2021, 50 years later, where this company in Silicon Valley called Cerebras, they built these highly specialized, deep learning processors. They have a staggering 2 trillion. Okay, so you go from 2,000, that's 2 trillion. That's a factor of 1 billion in 50 years. So, think about your investment. You invested $1 in 71, now it's $1 billion, right? I mean, where else, in what other field, have we ever had a factor of 1 billion over 50 years? So whether that's memory or CPU is absolutely essential core at the AI revolution. So now the big question is, can these deep machine learning networks do everything, or is there still something fundamentally missing? Clearly, they do it very different than the way humans learn, right? You don't have to see things 1 million times. You can abstract very quickly. I can show you something very new, something you've never seen before. You look at one instance and then you can immediately generalize. Children don't look at, millions of hours of training videos. So we're probably missing something. And can we learn that? Can computer science and AI still profit from learning the way humans or the way animals do things? Or is that all cashed out? Is it just, okay, we learned in the past, we learn neural networks come from, Hubel and Wiesel, McCulloch-Pitts, Rosenblatt, all of that, neocognitron. But is that, okay, that's history. Today, we don't need to worry about the brain anymore. We can do it all. And that's the big open question, whether that is true or not.
Well, looking into the future, do you have any feeling for what the role of a professional society should be?
Yeah, it should be, it should be to represent us to the general public, to represent us to Congress, to represent us, to engineers, people who want to make a career. So I think professional societies are an absolutely must. And I think, to continue to nurture the field, reach out into schools, reach out into the larger community, make us more diverse. Right now, we're not very diverse. All of that could be, is greatly facilitated by having professional societies like INNS. Absolutely essential, in fact.
Now, you've been a close observer of the whole AI field for quite some time. Do you have any feeling for what people are doing right or what they're doing wrong now in AI?
It's not for me to judge. It's, we have to wait 50 years and then look back to see, what were the mistakes made. I would caution people, however, I mean, right now you get triumphant voices that say, well, I think we've sort of solved intelligence or will very soon. And I think we are still far from that. And I think it behooves us to be a little bit more humble and you know, and again, go back to the inspiration, the only system that we know are truly intelligent, which are humans, and learn and still try to understand how we can learn certain things with such great ease. Now, of course, human intelligence, as we can see in the current political climate, you know, also has its limitation. The limitation of human rationality, right, is clearly apparent to everyone. But I think it's still, there's much that we can learn. And so I think AI should not totally turn away and say, okay, we've done, it's all going to be neural network, deep neural networks from here on forward. I still believe we're missing some significant new inspiration that might come from looking at brains or looking at the way brains develop, which is very different, of course, from the way we train computers. But that's just my intuition, partly because I'm a neuroscientist, of course. So it remains to be seen whether that's true or not. I mean, it's very impressive if you look at AlphaFold, you know, and it's not just one technique, it's not just deep machine learning, it's reinforcement learning, it's a lot of clever interpolation, how they can now, you know, for most protein sequences, they have the structure, and the structure looks pretty close to the structure when they actually do, you know, three-dimensional X-ray crystallography. So that's really pretty impressive. So, you know, the chat box again of, you know, Siri and Alexa and Lambda, the latest chatbot of Google, again, looks very impressive. Tesla is moving very rapidly towards, you know, totally autonomy. So we'll see what the next decade brings.
And that next decade will involve a lot of new researchers in the field. What advice would you give for a researcher just starting out in the field?
Oh, it's very exciting right now because we can solve these long-standing problems. Yeah, I mean, I would say on both sides, whether you're more engineer or more a biologist, you know, stay young, stay fresh, always question and look towards the other field. You know, as computer scientist, it might still behoove you to look towards the brain. And certainly as a brain scientist, we have to. I mean, we're forced to. I see it every day at my institute. We couldn't do all the stuff we have because we generate now petabytes or even more of certainly gigabytes per experiment and petabytes in some of those experiments for example, for the connectome, which are simply, I mean, no human will ever see this data. The vast majority of the data that we now generate will never be seen by a human. Right, so that requires deep machine learning to mine the data, for example, to look at the connectome data, to extract the actual connectivity. It simply can't be done by humans because it's far too laborious. So we totally depend in neuroscience on AI and learning algorithms. The question to what extent AI, in particular future development in AI, depend on brain science, that remains an open one.
Well, is there anything else which you would like to add to what you said?
No, I mean, there are many other questions. One could talk about computer consciousness, future ideas about computing, neuromorphic computing, but let's leave it at that.
Well, Dr. Koch, very much appreciate your spending this time with us and look forward to reading more about all the work which you'll be continuing to do. So thank you very, very much.
Thank you, David, for having me. I appreciate it.